So I would first like to thank the organizing committee and Ajinomoto for the invitation to come and discuss with you. And I think that the table was really nicely set up. Thanks a lot to the previous speaker. To a topic which is very dear to my heart is that are we ready to move to balanced diet for essential amino acid rather than MP or metabolizable protein, provided that we do meet the RDP requirements. So I would like to acknowledge the work of my co-authors, Roger Martineau and Daniel Ouellette from Agriculture and Agri-Food Canada, and Simon Bingali and Doris Pellerin from Laval University. So I just put those abbreviations so you have that into your papers. So actually, are we ready to balance diets for for essential amino acid rather than MP? But before that, and again, that was quite well presented before, is that I think we need to agree on one point. You know, if we want to balance the diet for essential amino acid, we have to quit balancing diets for CP. And we have to agree as a starting point that we're much better off to balance diets for MP rather than CP. And I do just have a few examples here where we do have the milk true protein yield on the y-axis. And here we have total MP supply, and here the crude protein intake. And the smallest is the better, so we see that we have a much better relationship when we look at milk protein yield in relation to MP supply rather than CP. Obviously, often we hear, well, CP is something we can measure. Yeah, but and MP, as being said before, we need a model. But as I'm often saying, it's better to be approximately right than to be perfectly wrong. So I think it's much better to work with the MP than the CP. And this is from a meta-analysis from Utanen and Ristov, actually looking at CP or MP as a predictor of milk protein yield. And again, the smallest is the better. So you can see that when milk protein yield was being related to CP, the relationship wasn't as good as MP. And there was a small improvement when a quadratic term was being included. So we start with that. We all agree that, you know, it's much better if we balance for MP than for CP. And the question that we do have is that, do we gain in efficiency if we balance for essential amino acid rather than MP? And the analogy I really like to use when I'm talking about amino acids is to compare the amino acid to the letters. In the ABC, we do have 26 letters. With those letters, you can write all the words, and with that, you can write all the books that you want. The same way, we do have 20 amino acids, and with that, we can build all the proteins that we need. That would be the milk, that would be the tissue, that would be the fetus, that will be the enzymes, hormones. It's all built the same way. And as it is for a word, you know, an amino acid cannot substitute for another one. It's just like if you're playing Scrabble, and I give you 100 letters random. Not sure how many words you'll be able to write with that. But if you tell me which letters you want, then you will need much less letter to be able to write the same number of words. And basically, all those letters that you can't use, this is basically the nitrogen that you will find under the tail of the cow that will be lost in the environment. So very briefly, 20 amino acids, 9 essentials, 1 semi-essential. I don't think we can treat arginine as we do treat the other essential amino acid, because there is really a substantial de novo synthesis by the cow for the arginine. And the 10 non-essentials, and what I wanted to point out with that is that these non-essential do require nitrogen to be synthesized. I don't think this is a problem that we will have in the dairy cow because, as it was said before, and I am not a rumen person, so that will be my comment for RDP, but the requirements for RDP really needs to be met just to ensure that we have adequate dry matter intake. So we do have also adequate rumen environment that will allow a good digestibility, that will allow a good milk protein yield, that will allow a good good microbial protein yield, that will allow also a good milk protein yield. In the pigs, for example, where they can make this very synthetic diet where they just added the essential amino acid, they came to the point that basically they were restricting the performance of the pigs because they were giving all the essential amino acids that were needed, but here, the animal needs to be able to make those amino acids. They can make it, but they need the nitrogen to do it. So what they did is that basically they just added ammonia to the diet that was made only from essential amino acids, and after that, the performance of the animal will be restored. But I don't think this is a problem that we will face with the dairy cows because, again, it is very critical that we do meet the RDP requirement just for the function of the rumen, but doing so, we will provide enough nitrogen for the synthesis of the non-essential amino acids. So if we want to balance the dairy rations for essential amino acid, we do need to move to balanced diet using a factorial approach. In NRC 2001, well, that were actually the specification that we're being told that we need. So we need to know the net protein requirement for maintenance, growth, pregnancy, and lactation. We do need to have a good amino acid composition of these different functions. And we need, this is very critical, to know the efficiency with which these absorbed amino acids will be used. So hopefully the next NRC will be written before the next 10 years. We're working hard on that. And, but this is a challenge, actually. It's, I've been saying that we need to move with variable efficiency, for example, but putting it in a model is something different. So at that time, at the NRC 2001, it was the opinion of the subcommittee that the current knowledge was not available to move to a factorial method. But the question is that, what have we learned since 2001 that would allow us to do that move? So to look at that, we'll look together at, is it MP or is it essential amino acids that do drive milk protein yield? Then if we want to balance for individual amino acid, we'll look at general considerations. And when we balance, basically what we want to do is to match the supply with the requirement. And we'll see at the end what will be the economic and environmental impacts of balancing for essential amino acid rather than MP. So I've put together a few studies, there isn't that many, where we do have a different MP supply, but we do have a similar milk protein yield. And often those aren't studies that are just with dietary changes, because usually when you make a dietary change, if you increase crude protein, if you increase MP supply, you will increase most of the essential amino acid supply. So this is a study here where we had cows that were fed a low protein diet, and then that would infuse with non-essential amino acid. And what you can see is that milk protein yield, MP supply increased, but milk protein yield did not. The same study where the cows were then infused with essential amino acid, the non-essential amino acid was being added, and what you can see, again, MP supply did increase, but milk protein yield did not. Here, a diet that was MP adequate, MP deficient, but with lysine and methionine and histidine as rumen-protected amino acid being added. And what you could see is that MP supply did decrease, but milk protein yield didn't change. So obviously you can alter MP supply, but doesn't mean anything in terms of what will be the prediction or what will be the response of the cow to, in terms of milk protein yield. Here, Jalongo, the same type of study, MP adequate diet, MP deficient diet with lysine, methionine, and histidine added as rumen-protected amino acid. And what you could see is that not much change in terms of milk protein yield, despite the fact that there was a clear decrease in MP supply. And finally, a study from France. Again, MP deficient diet with essential amino acid, MP adequate diet, but this time non-essential were being added. Again, an increment in MP supply, no change in milk protein yield. So if we put that together, we do have here milk protein yield and MP supply. Overall, it's not that bad. But if you look within studies, we do have a few studies basically where milk protein yield decreased despite the fact that we had an increment in MP supply. And why would that be that way? Well, actually, I put here the milk protein supply, and I put here on the y-axis the sum of histidine, lysine, and methionine. And what you could see is that for some studies, especially here and here, despite the fact that MP supply increased, histidine, lysine, and methionine did not change or even decrease, because here, there were some that were being supplemented to the protein-deficient diet. So if we redo the graph, and rather than put, if we redo the graph, and rather than putting MP supply here, we put the flow of histidine, lysine, and methionine relative to milk protein yield, except that one here, and there might be an overestimation of the supply of methionine. Well, you can see that within study, at least, we don't have the negative relationship that we have had before. And I will tell you later, you will see why I've picked the sum of histidine, lysine, and methionine and couldn't do like 10 graphs, but you can see that when we relate to these amino acids, actually this is quite a good relationship. And actually, I just saw yesterday in the GDS alert, there is a meta-analysis that had been published with, there was Santos, Charlie Sniffen, Ken Griswold, where they look at the metabolizable amino acid supply that were estimated by CNCPS versus milk protein yield and milk yield. And what they reported was that there was a clear relationship in terms of the percentage of milk protein with the methionine supply, and milk yield was with methionine and histidine, and also leucine and threonine. They didn't find any relationship with lysine, but they were saying that it might be that the range wasn't that changing, and also maybe it was a little bit too low. But well, it just impressed from GDS yesterday. So I thought to make a slide, but I just couldn't make it. Actually, with these studies, I think that it's quite clear that what is driving milk protein yield is not per se the total MP supply, but it's more like critical amino acids. But it's a little bit hard to get that when we have changes from dietary supply. And here I just wanted to put the MP supply here, and here histidine, lysine, and methionine. And obviously, when we make dietary changes, well, obviously, these are moving together. So it's kind of hard compared to what we did have here, that we have MP supply and is, lys, and met that are not going the same direction. But again, it's not that easy to make the relationship, well, to exclude MP supply from histidine, lysine, and methionine. So really need to go through very picky studies that have been carefully looking at that. So if we want to balance the rations for individual amino acids, the first thing that we'll go through is that general considerations for the amino acid composition of protein. Actually, when we were working at the way things were being used and balanced, to balance for essential amino acid, the one thing that we looked at is that how was the amino acid composition being determined? And when we looked at it, basically there's something that just jumped to me is that when we were looking at the supply and all the requirements except milk, everything was being from an hydrolysis of the protein matrix with the 24-hour hydrolysis. Whereas for milk, in most of the program, actually, the amino acid concentration was obtained from the DNA sequence that was obtained from the different milk protein fraction. And basically, when you want to match that, because when you want to match the supply and the requirement, you're adding together all the amino acid from the supply, which will be all the microbial crude protein plus the RUP. You want that to equate to the different protein functions that are the maintenance and milk protein, basically. And then you have like the requirement calculated one way, part of the supply calculated one way, and part of the requirements calculated another way. So it's just like trying to add together bananas and apple. So we try to put all that more uniform. And actually, it is something which is not new, but it has been somewhat forgotten, that the standard 24-hour hydrolysis of a protein is not something perfect. It is a compromise. Basically, it's not too bad for every amino acid, but it's not good for, it's not really precise for any amino acids. And here you will see why. This is an example of the concentration of valine in canola meal. And what you can see, here it's over time. So we have here the traditional 24-hour. So we can see that after 24 hours, there was still release of the valine, after some time. Whereas for other amino acids like serine and threonine, they were all released actually after 15, 18 hours. So when we wait to 24 hours to do the measurement, it's already beginning to be degraded into the hydrolyzate. And so to determine really what is the concentration at time 0 in the protein matrix, basically we need to run, but we won't have to do that all the time, but just to put the factors and get the numbers, you need to run that with, here it's like 13 different times. And with that, what we did is that we propose still to continue to use the values that we obtained in 24-hour hydrolyzers, but to apply to those some correction factors. And what we did is that we did compare different matrix, like duodenal samples, fecal samples, different feed samples, also feed that had been incubated into the rumen for 18 hours, and we couldn't find any difference in the recovery of the different amino acids. So maybe, after more and more analysis, we'll get something, but for now, we propose just a correction factor, which is going to be the same for all the amino acids. The other point that we've forgotten is that when you hydrolyze, I know it's a little bit technical, but it's really important to get the basis, because when you hydrolyze a protein source, basically what you do is that you cleave like the peptide bond between two amino acids. When you do so, you're adding a molecule of water to each amino acid, because when you do the protein, when you put two amino acids together, basically there's one molecule of water which is being released. So when you cleave that, you get one molecule of water which is being added. And what it means basically is that if you hydrolyze 1 kilo of pure protein, that will not yield 1 kilo of amino acid, that will yield between 115 and 117 kilo, 1.15 to 1.17 kilo of amino acids. So you will see later what is the importance of that when we, for example, are looking at the amino acid composition of microbial protein, because we don't want to have the sum of all the amino acid yielding 100%. We want to have the sum yielding 1.15 or 115%, because we've been adding this molecule of water to each amino acid that has been hydrolyzed or that has been released. And that is really changing the composition of the microbial protein that we are estimating. So again, just to, if we put the molecular weight of the anhydrous amino acid on the molecular weight of the amino acid, plus what is missing in the 24-hour hydrolysis, this is giving us a global correction factor. And this is really what we need to have to determine the true protein on the crude protein ratio. And just to tell you where this is bringing us, actually this is from the paper of David Pacheco, who was a postdoc with us, and we did compare the histidine net portal absorption with what's being predicted to be digested by NRC, and that's really where we began to wonder what was going on, because in theory, you cannot have more amino acid being absorbed in the portal vein that you have being digested, because we know that there is some catabolism of amino acid by the gut. Some will be used to support the metabolic fecal output. So then we're beginning to look at what would be the reason for that. And one of the reasons, well, would be this correction factor that needs to be added. And when we add it here in blue, you can see that it's not yet perfect, but at least, we're getting closer to what's being absorbed compared to what was first being predicted just by NRC without any correction factor. So the general consideration on amino acid composition for the first section, well, when we do have amino acid obtained from hydrolysis, correction factors really need to be used. We can, for now, use the same correction factors for all different protein matrix. And 1 kilo of true protein, and we'll see the example how it is changing things a little bit later on, should yield between 115 and 117 kilo of amino acid when it is true protein. The second point, when we want to balance the ration, actually, what we want to do is to match the supply and the requirement. So the supply is going to be the sum of the microbial, undegraded, and endogenous supply. And all of that will be digested, and this is what forms the metabolizable protein, or from what is being calculated, the amino acid digestive flow. We will focus first on the amino acid composition. So this is a paper that just came out recently. And actually, most of the model that we're using factorial approach so far, what they were giving as an amino acid composition to the MCP was what was published by Clark or by Le Inaf, or, so these were basically bacteria that were associated with the fluid phase, and there was no protozoa that was being taken into account. So with Michael, we put together a review. We wanted to do a meta-analysis to see if there was an effect of the diet on the amino acid composition, but there was not enough data on amino acid composition with different diets. So what we did at least was put together a review where we had the amino acid composition of the fluid-associated bacteria, the particle-associated bacteria, and protozoa. And you can see that, for example, lysine is about 50% higher in protozoa than it is in the bacteria. And this, with this proportion here, we came to what would be a composite value of MCP. I know that in the next version of CNCPS at 7.0, they want to have a model that will account for variation in the proportion of the protozoa and the bacteria, but for now we just have a fixed effect. And I put just beside, in green here, what is from Clark and what is from Le Inaf, which is the basis of the French system in green, in yellow. But this is gram of amino acid per 100 gram of amino acid. And as I was saying, we have to think that true protein will not yield 100 gram of amino acid. It will yield 1.15 grams. So if we report now the grams of amino acid per 100 gram of true protein, you can see that it is really increasing substantially the composition of MCP for each amino acid, especially lysine more because now protozoa are being included. And now, if we come back to this comparison that we did, where we had net portal absorption here in green, we have in yellow what was predicted by NRC being digested, and now we have what is being predicted by NRC but corrected for the amino acid composition of the bacteria, of MCP, I should say, plus what is being lost in the 24-hour hydrolysis. Now we can see that at least we begin to have predicted flow of what is being digested, which is higher, at least for some amino acid, than what's being absorbed, which is making more sense biologically. For the RUP, actually, what we do suggest is to use still the same correction factors. As I said, we couldn't find any difference in the correction factors for after a 24-hour hydrolysis for samples of feed that had been incubated into the rumen. And the last point, and if you've heard me before, you know that, what's my thinking about that is that, when we're talking about the duodenal endogenous nitrogen, is it a supply, is it a requirement? So what is those endogenous secretion? Basically, those are secretion. Again, this is my usual scheme. So you have the rumen, oomesum, abomasum, small intestine, large intestine. We have here, the blood, which is pumped by the heart, freshly oxygen, which is bringing all the amino acid, all the nutrients to the gut, and we have here what's being drained. And basically, what we do have is that we have those amino acids that are being brought mainly from arterial supply that are being used to synthesize the endogenous secretion that will be secreted into the lumen of the gut that will be flowing at the entrance of the small intestine. Part of that, as we can see here, will be digested and basically returned. So if we look at the more global picture, we will have endogenous secretion that will be flowing all the way through the gut. Part of that will be digested, and what is not digested actually is a true loss. But what is being digested and just returned actually is not a true loss because it's just a recirculation. So actually, what we're saying is that it should not be included as a net supply. So it needs to be removed from the duodenal flow when we do the measurement of the duodenal flow. But it's not a requirement neither, because if I go back, if I get some amino acid flowing here, they are absorbed. This is not a net demand for the animal because it just returned to the animal, so that shouldn't be included into the supply, neither into the requirement. So the second part of the amino acid, if we want to balance for amino acid, actually, for the supply, we do have the microbial, to which we want to give an updated amino acid composition, which is taking into account what is missing in 24-hour hydrolysis and the true protein that is being obtained from hydrolysis, the undegraded fraction, and we want to remove the endogenous fraction. What is being digested will now be MP and amino acid digestive flow. For the requirements, how I see that is like basically two aspects. What we need first to define is the amount of protein, and before it was more called like the net protein requirement. So the amount of protein or the amount of amino acid that needs to be replaced because it's being secreted out of the animal, or it's going to be in accretion, either in the fetus or body protein gain. But I will just keep it simple, so we will have a cow which is not in gestation, which is not changing body weight. So we have the different functions, basically, which will be scurf, metabolic fecal, endogenous urinary, duoendo, who will cross it, and milk. And to these functions, we need to apply an efficiency of utilization. Because if we need, for example, that would be for MP, or that would be for amino acid, if I have 100 grams, for example, of lysine which goes into milk, how many grams of lysine do I need to digest to support that function? And that will be given by the efficiency. So we really need to tackle this efficiency, and this is really a critical issue because that will be finally the driving force. So the export protein, we'll have an example for a cow which is 45 kilos of milk at 3% of true protein, 27% kilo dry matter intake, no growth, no gestation. We cross the duodenal endogenous. So this is more detail in the corner presentation I gave last year, but basically scurf, we're taking back what is being presented by Swanson, but we give a true protein to crude protein ratio because it's minimal actually in terms of the total requirement. But what we want to have is something which is really systematic across all the different functions. We have a crude protein exportation. We transform that into a true protein export. Then we assign to that an efficiency, and then we do have the requirement. So the scurf, basically this is Swanson, but with a true protein to crude protein ratio of 85%. Endogenous urinary, it has been increased compared to what is presented by Swanson. And this is based on the amino acid, the different protein fraction or the different nitrogen fraction that are present into urine. Metabolic fecal protein, actually in terms of export, it is much smaller than CNCPS or NRC. But actually, if you look at CNCPS or NRC, there is no efficiency attached to this fraction. So we wanted to have like all the proteins which are being secreted out of the animal treated the same way. So, and this is based on work that we've done with N15 labeled leucine and adapted from an equation from Marini because we wanted to have somewhat of an impact of the quality of the diet. So this is the requirement for metabolic fecal protein. And milk, basically, this is based on milk true protein. So if you compare the export proteins compared to what is in NRC 2001 or CNCPS or the proposed update, scurf, as I said, it's really minimal. You can see that the endogenous urinary, as I said, is largely increased. Metabolic fecal, this is just the net export. This is smaller, but again, we will attach an efficiency to that, whereas NRC and CNCPS, there is no efficiency. The endogenous duodenal was only into NRC, so nothing in CNCPS and in the update, and milk, which is fairly straightforward. The amino acid composition in those export proteins, again, we need to express that in terms of the true protein, and that need also to be corrected for what is missing in the 24-hour hydrolysis. So we do have something which has been consistent, and the amino acid composition of milk has been established based on a review of the different protein fractions that are into milk. So this is for the export. But what is really new and that we really need to tackle is the requirement. Either for amino acid or for MP, traditionally, we have had the the requirement that was defined. We had the export for maintenance divided by an efficiency of maintenance, and the export for milk divided by an efficiency of milk. And this has been changed recently in Van Amberg 2015, but a good example of that, for example, was before in the CNCPS. They had, for example, for methionine, an efficiency that was, I think, 85 for the maintenance, and that was 100% for milk. And the requirement was the sum of the two. And what I want to share with you is how the studies that we've been conducted on amino acid metabolism can help us to define better what would be these efficiencies and how we can apply that. So again, the same scheme as what we've been presenting before. So we have rumen here, the omasum, abomasum, small intestine, large intestine. We have the blood, which is coming from the heart, which is being drained. Here we have all the mesenteric vein, gastrosplenic vein, which is all getting together into the portal vein, flowing into the liver. So what we do is that we put a catheter in one artery here, which is draining the gut in the portal vein. So the difference between what is flowing out of the gut and what is flowing in will give us what is going on into the gut. When we're interested in two amino acid, that will give us the flow of amino acid, which is absorbed, from the digestion of protein across the small intestine. Plus, actually, when we have the portal vein here, that's going to be the summation of what's being digested across the portal vein, across the small intestine, plus the use across the whole tissues. Here we put a catheter also in one hepatic vein. So again, difference between portal, what is getting into the liver and what is getting out of the liver will give us the liver metabolism. And we also take a sample at the exit of the mammary gland. So we do have altogether the major amino acid users in the tissue, in the whole body. Actually, about 45% of the protein synthesis, not the protein exported, but the protein synthesis in the whole body is being made by these tissues, despite the fact that they only represent less than 10% of the body mass. The same thing for the energy. So I know I'm a little bit biased, but actually total splanchnic tissue, the liver and the PDV, this is less than 10% of the body mass, but it is using close to 50% of the energy which is being used by the cow. So when I'm talking to dairy farmers, I'm saying, well, your cow which is being eating like between 25 and 30 kg of dry matter, think of that. You get close to half of that, which is just being used for the turnover of these tissues. And to show you how active it is, there is about 2,000 liters per hour. You can put that in US gallons. That's going to be like 500 US gallons, a little bit less Canadian gallons, that will be flowing out of the liver. So that this is really showing you the really intense metabolic activity across these tissues. So if we look here at, and this is from all studies that I get at that time in 2012, and we will concentrate on lysine and methionine. So we have like the lysine flow here, net portal absorption relative to what is being digested. So we have still a little bit which is not appearing in the portal vein because it's used to support metabolic fecal output. But what you can see is that there is very little lysine removal by the liver. So the post-liver supply is almost equal to the portal absorption. And we do have a mammary uptake which is smaller than the post-liver supply, which is telling us that there is some catabolism of lysine in peripheral tissues. And we have a milk output which is smaller than the other uptake, which is also telling us that there is some catabolism of lysine within the mammary gland. If we look at methionine, we do have a totally different pattern. We have here the net portal absorption, which is slightly lower relative to net digestible because, well, that was based on one study in sheep where there was oxidation of methionine across the gut. But what you can see is that there is a fairly important removal of methionine across the liver, so that the post-liver supply is smaller, much smaller than the portal absorption. And basically, we do have a mammary uptake which is equal to milk output. Well, actually, I put all the studies that I had that were published at that time. Often what we do have is that the post-liver supply is fairly equal to milk to mammary uptake, which is pretty much equal to milk output. So here, this pattern that we have no liver removal, post-liver supply greater than mammary uptake, greater than milk output, is typical from what we call the group 2 amino acid, which is together the lysine and the branching amino acid, isoleucine, leucine, and valine. Whereas this pattern here, where we do have substantial hepatic removal, a mammary uptake which is equal to milk output, is typical of the group 1 amino acid histidine, phenylalanine, and tyrosine plus tryptophan. So what is telling us that those data? Well, I call it like amino acid catabolism is somewhat geographical or anatomical. Is that doesn't matter how much methionine you will give to the animal, how much protein you will give to the animal, the mammary uptake will always be equal to the milk output because there is no enzymes into the mammary gland to catabolize the methionine. So then there is no reason to call an efficiency of lactation of 100% because basically all the catabolism of the methionine, even if you give more protein and if you give more methionine to support the milk output, catabolism will never occur across the mammary gland. Doesn't mean that there is not like inefficiency of lactation for this extra protein or this extra methionine that you give to support milk protein yield, but it will not occur within the mammary gland. So therefore, what we did is that we proposed to use what we call the combined efficiency. You know, if all the catabolism of the group 1 amino acid occurs within the mammary gland, why should we try to define an efficiency for the metabolic fecal output or for the milk? Why should we just use like a combined metabolic fecal, a combined efficiency? Because basically it will all occur biologically at the same place. So from the database that I've been presenting before, we looked at the mean combined efficiencies. What we did is that we put all the export proteins and then on the y-axis all the supply, and then we made the division and we looked at what was the mean. And we had another database that has been published in Martineau et al., where what we had, we did the same exercise, but rather than having diets that were supplying extra MP, we had only casein infusions. And we compared to what is being used by CNCPS that was proposed from studies also that we had a few years ago. And basically what we could find is that, you know, on average, it wasn't that bad. It's a little bit higher here. So I do wonder if with the diet database, if we do not have more limiting diet, and methionine here, which is higher in terms of the mean efficiency. But what I want to raise your attention to is that it is the mean. What it means, actually, is that it is, on average, what you obtain. But here, you might see it better on your notebook, but this is for all the amino acid, and you can see that it is highly variable. You have the mean, yeah, but what you can see is that it is really changing depending of, that's what we want to look at, but it is really not constant. And there are 3 amino acids, it's going to be histidine, lysine, and methionine, that the mean efficiency is really close to 80% or even higher than 80%. If we go back here, you see histidine, lysine, and methionine, we're in the range of 70, 79%, whereas for another amino acid, valine, we were below 70%. So I think what it is telling us is that I don't think it's because the cows are less efficient in using methionine, histidine, and lysine. I just think that because of the profile of the microbes and because of the profile of amino acid in the diets that we're feeding the cows have to use that more efficiently. And I think this is the reason why we do have, as I presented at the beginning, a good relationship between histidine, methionine, and lysine compared to the other amino acid, because the other amino acid, they have much lower efficiency. So it's like from the microbes or from the diets, they're somewhat like relative to the other amino acid, a little bit more in excess. And just one point that I wanted to make is that it is true that microbial protein are really good in terms of the profile for lysine and methionine. However, if you continue the comparison, if you look at histidine, you can see that histidine concentration is microbe. It is fairly low. And this is some work that I see Alex in the back, but that we've done with Alex and some of the first studies that I presented, that when we began, we decreased the amount of protein that we were feeding in the diet and supplemented with rumen-protected methionine and rumen-protected lysine. And when we did that, there was, we were not able to restore the milk protein yield. But when we looked at the data, and the one thing that was really striking was the very low plasma concentration of histidine. So we said, wow. And then, we began to look back and we said, yeah, the hypothesis was that people from Scandinavia, they have been looking at infusion of histidine, and they found that the cows were responding. And we said, oh, well, it's like with barley silage or with grass silage that histidine might be deficient. But actually, it's not the type of diet. It is just that with this type of diet, the protein supply was relatively low, and therefore, the contribution from the microbes was higher. So if you have a great proportion of your supply of MP, which is arising from the microbes, then the proportion of histidine will be smaller. And this is really one of the amino acids that we have to pay attention to, especially in a context where we want to decrease the total protein that we're feeding the cows. So this is bringing us to the variable efficiency. So before, the idea was that most of the models were working with a fixed efficiency. So the return into milk from an increased amino acid or protein supply is identical until you reach a breaking point. And we really want to move that to a variable efficiency. So as you do increase the supply in lysine here, you can see that for an identical increment in the supply, you decrease here the return into milk compared to a lower supply. First, our idea was, well, it was quite neat when we began to do that the efficiency was related to lysine, to the amount that was being supplied. And just to show you how, again, variable it is, this is the average that we presented first, but you can see the minimum and the maximum. The maximum would imply that the cows are using their body reserve because, obviously, they can't make those essential amino acids. If we plot together this combined efficiency relative to the digestive flow, again, from the database that I presented initially, we can see that effectively it does decrease as the flow does increase. And this is, for the average, it's quite a wide range. Then we begin to think a little bit more, and actually, if we look at what's being used in the monogastrics, or even what's being used in the different European systems, going to be the Dutch system, the Scandinavian system, or the French system, they do relate the efficiency not only to MP or to amino acid, but to the ratio of MP to net energy supply. So now I just plot again the combined efficiency of lysine utilization, but rather than having just the digestive flow, this is the digestive flow related to the energy. Basically, somewhat similar to what is being done in the monogastric. And what you can see here, this is really narrowing the range at the mean efficiency. And we have here, which is showing that we do have a better relationship than what we had when we only had the digestive flow. So actually, this is making sense. It's kind of known that if you increase the energy supply, you will also increase the efficiency of utilization of the amino acid. But it's really how we're going to put that into the model. Actually, my friends, statistician friends, I like it. I'm simple. It's a nice graph. They don't like ratios. So they said, well, Helene, you have to put that in two different independent variables. So just to show you, again, this is just really fitting to the measured efficiency. But if we now use the calculated efficiency with the equation, what is interesting is that you can, what is interesting is that you can see that effectively, right, you can get a 70% lysine efficiency with different lysine supply, but that will be affected by your energy supply. So I think that we're really heading into the right direction. We might not have it perfectly perfect, but we're getting there. So the requirements, again, we need to have the export as true protein. We have to correct the amino acid concentration, so we consider what is missing in 24-hour hydrolysis. We want to work with a variable efficiency. Work in progress, work in progress, so it's not perfect, but I think it's better than fixed efficiency. And what I'm always saying is that, we can always say, okay, it's not perfect, I don't do anything, or you can begin and work, and, at the end, we're going to get there. So I wanted briefly to touch at what would be the economic and environmental impacts that was assessed by, with using the N-cycle. This is a software that had been put together by Doris Pellerin, Michel from Laval University, and Michel Watsio from Wisconsin. And this is an Excel-based linear model. And what we did, it was based initially using MP model. And, it was based using the NRC model. So it is referred to as MP 2001. And then we looked at different propositions that I've been presenting, looking at the amino acid revised recommendation. And this is AA revised. Sorry, it has been done in a Canadian context, but I think that tendency will be the same if we transfer it to what's being done in the United States. So we had the Maritimes. So you can see here the average. This is based. That was based on data on. That was based on data that were collected on the farms. So average between 2010 and 2014. Good time for us. This was the exchange for the Canadian versus the US dollar. It is quite different now. So we have Maritimes, Central Canada, and the prairies. So different here, more forages. Here we have more, maybe something closer to the American system with corn silage and soybean meal. And here we have a fair bit of wheat, distilled grain, and barley silage. The requirements, based on what I've been proposing before, and the supply basically was reassessed using MCP estimation from White et al., the microbial composition from Suck et al., feedstuff that was corrected for the 24-hour hydrolysis, and for growing animal, we used the Beef NRC 2016. And this is the net income. This is in Canadian dollars per kilogram of fat and protein corrected milk. And this model, what it's doing is not only looking at the revenue, the increased income that is coming from milk. It is really on the whole farm basis. So what is interesting is that independent of the type of system that we're using, we always see when we balance for amino acid rather than MP, we always see an increment in the net income. And the same way, this is not depending, of your starting point. Your imbalance here with more forage was a little bit lower. But when we balance for essential amino acid, and again, that was only for histidine, lysine, and methionine that it was balanced, we do decrease the nitrogen balance per kilogram of fat and protein corrected milk that was being produced. So in conclusion, if we want to balance for essential amino acid, we need to base our requirements on the export true protein. If we want to be homogeneous, we need really to do a correction for the values that are obtained with the 24-hour hydrolysis. If we want to add that to the amino acid composition of milk, which is obtained with the DNA sequence. Again, we had to, especially it's very important for the microbial protein, we have to have the amino acid corrected, but based on the true protein. And we can put together a combined and variable efficiency. And with correct assumptions, actually, what would be nice is that the factorial method should work for all essential amino acid, question mark for arginine. This is increasing the net income. This is decreasing the nitrogen balance. And the question is, are we ready to forget about MP and balance only for essential amino acid? And I'll put here RDP because I think that we need to feed the cow, but we need to feed the rumen bugs. And I want to thank everybody that has been helping us and supporting us in our work. And questions will wait. And I do thank you for your attention. Thank you.